Welcome to Cell Biology. This topic is the introduction to GCSE Biology. It is crucial to understand this topic well in order to really understand the subsequent topics. It relates to everything, it flows through very, very nicely. So we shall begin. Firstly, all living things are made of cells, and when we say living things, we include plants and animals in this. Often people forget about plants being part of this, and also bacteria falls under living things, which is why we'll see plant cells, animal cells, and bacteria cells shortly. Broadly, the cells fall into two main categories. There's prokaryotic and eukaryotic. We'll start with prokaryotic because it's a little bit easier. They are small and simple cells. Bacteria is the only example of prokaryotic cells that you really need to know about. They are single-celled organisms, so the entirety of one bacteria is one cell. Whereas when we move on to eukaryotic cells, they are more complex, and they're your typical plant and animal cells that we will come across shortly. All of these cells contain parts called subcellular structures, so your nucleus, your cell membrane, all of the parts that are inside the cell fall under subcellular structures. So we will start with animal cells. Animal cells contain 5 parts that you need to be able to label on a diagram, and also know what each part does. So that's nicely outlined here on this particular slide for you. So we have the diagram on the right over here. We have the nucleus in the middle of the cell. The nucleus contains genetic material. Genetic material, you could also say, just contains DNA. DNA is the genetic material that makes up all plants and animals. It contains all of the information about that particular organism, such as, for example, for humans, eye color, hair color, all of your genetic characteristics, any illnesses, etc. The cytoplasm is the liquid inside the cell. If you get a question asking you about the function of cytoplasm, you need to explain that it is where most of the chemical reactions take place. Okay, as it says here, it contains enzymes that control these chemical reactions. The cell membrane, going around the outside over here, holds the cell together and controls what goes in and out. We will see a lot more about the cell membrane later, especially when we look at diffusion and osmosis and how things get into and out of the cells. The mitochondria are very, very important to understand. They are where the reactions for aerobic respiration take place. Now, aerobic respiration is how a cell releases energy. We will again come across respiration in a lot of detail in a later topic, and this is why all of these things interact very nicely. You need to understand that cells which require lots of energy will tend to have lots of mitochondria so that they can release more energy for themselves. And lastly, ribosomes are where proteins are made in the cell, and again we will come across what kind of proteins a cell may need to make as we look at later topics. So these 5 subcellular structures are very important to be able to label if you need to on a diagram, and also if you get a question about what the function of each of them are, here it is outlined for you on this particular slide. Let's now move on to plant cells. Plant cells contain the 5 subcellular structures that we had from before, plus 3 more. It's often easy for a lot of people to remember it in terms of the numbers, so you don't forget, you don't miss any of them. So if you remember, there's 5, the same 5 parts again, plus another 3. Those extra 3 parts are the cell wall. So around the outside of the plant cell, we have an additional layer. That is the cell wall. So the cell wall is on the outside, the cell membrane is further inside. The cell wall strengthens and supports the plant cell. That's essentially what you need to write if you get a question asking you about the function of a cell wall. It is made of a substance called cellulose, which again we will come across a little bit later. We then also have inside the middle over here a permanent vacuole, which contains a substance called cell sap. It's a sticky substance of sugar and salts. And lastly, probably the most important part of a plant cell is the chloroplasts. These are always or often labeled green on a diagram because they contain a green pigment called chlorophyll, which helps the plant absorb light in order for it to carry out the function of photosynthesis. Photosynthesis is a process carried out by plants to help it make glucose or food for itself. Animals don't do that process, which is why you don't find chloroplasts inside animal cells. So again, all of these functions are important to have learnt and be able to recognize if any of those particular things are not labeled on a diagram, so that you can do that. We now move on to bacterial cells. Bacterial cells are prokaryotic, as we saw earlier. They are small and simple. The key difference is with a bacteria cell that it does not have a nucleus compared with animal cells and plant cells. It still does, though, have some of the things that the nucleus contains. So the genetic material, the DNA, is floating around inside the cell rather than being contained within a nucleus. In addition, the bacteria cell also has a cell wall, and it can have these rings of DNA called plasmids as well. Otherwise, it's quite a simple structure. It doesn't have the ribosomes and the mitochondria and the chloroplasts, etc., that other cells do have. The next part of this topic in cell biology is microscopes. Now, microscopes are often used, of course, to see these very small structures inside cells, or to see the cells themselves. And there's a little bit that you need to know about microscopes. There is a practical involving microscopes, but we look at that in a separate video. However, in terms of the details around some of the microscopes, they are listed on this slide. So the two common types of microscope that you need to be aware of are light microscopes, which are probably the most common ones that you've seen inside your school, the ones that you use in class. More powerful ones are electron microscopes. So light microscopes allow you to see the individual cells. The next part of this topic in cell biology is to do with microscopes. Now, there is a practical involving microscopes, which you do need to know about, but we look at that in more detail in a separate video. This slide shows you the theory behind microscopes that you need to know. So you will have used microscopes in class. Most commonly, you will have used light microscopes in class. They help you to see individual cells, so you can see the actual cell. For example, you will do a practical involving onion cells. You will see some of the cells, and you'll see some of the larger things inside the cell, like the nucleus. Electron microscopes are more powerful, essentially. Now, you need to be able to say that they have a higher magnification, which means that they can zoom in further, and they have a higher resolution, which means that they give you a sharper image as well when you zoom in. Electron microscopes help you see a bit further, so further inside the cell, or more detail inside the nucleus or inside the mitochondria. They take you a little bit further into the cell. So that's the key difference between light and electron microscopes. Electron microscopes have a higher magnification and a higher resolution. We now move on to talking about cell differentiation and specialization. A specialized cell is one that performs a specific function. So most cells in a human, animal, plant are already specialized. They form a specific function. So most of them actually look quite different to the cells that we saw previously. They have changed in order to carry out a specific function. Now, the word differentiation is just a word to say that a cell has changed to become specialized for its job. So an undifferentiated cell is one that looks a little bit like the ones that we saw earlier, so it's kind of in its original state. When it has differentiated, it means it has changed to a specific type of cell, such as a white blood cell. So it's a very small point, but it is something that you should be aware of, that cells do change from one type to another. And there are a few examples in GCSE Biology of specialized cells that you should be aware of. The first example that we see here is a sperm cell. Now, you often need to explain how this cell is adapted for its function, and explain how that helps it do what it needs to do. So firstly, a sperm cell, the key point about a sperm cell is that it needs to swim towards the egg, and then it needs to try and fertilize the egg. So there are various things within the sperm cell that specifically help it do that job. So the most obvious of those is, of course, the tail. It has a long tail. It allows that cell to swim from one place to the other. It also has a streamlined head, which again provides the ideal shape for it to swim. We often talk about swimmers having streamlined bodies that allows them to swim more quickly or more easily, and the sperm cell fits into that. The head of the sperm cell contains enzymes which help it digest the egg, or break through to to fertilize the egg. And of course then it contains the male DNA, which then combines with the female DNA to produce an embryo when it fertilizes. Another example of a specialized cell is a muscle cell. Now, a muscle cell, its main function is to contract, to allow you to move, to lift things, and so on. So there tends to be, or it tends to need, lots of energy. And as we saw earlier, the mitochondria are the key structures within a cell where respiration takes place. So they allow the cell to release energy. So you will find that muscle cells have a lot of mitochondria, because that allows it to release a lot of energy, and then that helps you move. And then there is also a plant cell that you need to be aware of that are specialized. A root hair cell, its job is to absorb water and minerals from the soil. Now, it wants to be able to absorb as many of these things as it possibly can. And so an ordinary cell like that, things need to absorb through the cell membrane. So water, minerals, they go through a cell membrane. If we can have a larger cell membrane, or a cell membrane with a larger surface area, that increases the amount of water and minerals that that cell can absorb. So this particular shape that a root hair cell has allows it to absorb water and minerals. Now, the other thing that you will notice about the root hair cell is that it doesn't have any chloroplasts, because roots are under the ground. Chloroplasts contain chlorophyll, which help absorb sunlight. Root hair cells won't get any sunlight, and therefore they don't need to have the chloroplasts. So that's how some of these cells are a little bit different to the cells that we saw earlier, because they have a specific function and they are almost adapted to carry out that function. Stem cells are undifferentiated. In other words, they're the stage before a cell specializes into one of these cells. So a stem cell is called an undifferentiated cell, because they have not yet changed into a specific type of cell. They have not become specialized for a particular job. They can divide to produce lots more undifferentiated cells as well, and that actually means that they are very, very useful for scientists. So this diagram here just shows that a stem cell can do two things. It can replicate itself, so we get more stem cells, or it can differentiate into a specific type of cell, in this case a white blood cell. There's two places where we find stem cells. Early human embryos, so when an egg has been fertilized, that initial stages of an embryo, the cells are undifferentiated because they haven't yet become specialized into the blood cells and the muscle cells, etc., that person is going to have. The other place where we can find stem cells are in adult bone marrow. Now, you just need to know the differences between the stem cells found in these two places. So stem cells that are found in embryos can turn into any type of cell, and that makes them particularly useful in medicine, as we will see a little later. The stem cells that are found in adult bone marrow can only develop into certain types of cells, e.g. blood cells, so they're not quite as flexible as stem cells found in embryos. This takes us nicely onto why stem cells can be so important in medicine. So the stem cells that we find in embryos, as we said, because they can turn into any type of cell, we can use those stem cells to replace any faulty cells in people who aren't well. There's a process called therapeutic cloning, where we make an embryo which has the same genetic information as the patient, the person who is sick. So the embryo will produce stem cells which contain the same genetic information as that patient. This would mean that because the stem cell has the same genes as the patient, or the stem cells from this embryo, they wouldn't be rejected by the patient's body. Now, rejection happens when your body thinks that those cells are foreign in some way, and so it starts attacking them. It thinks it's like a virus or so on. But if we have the same genetic information, then that shouldn't happen. And so what we can do with this method is turn those stem cells into cells that are useful for that particular patient. For example, we can produce nerve cells for people who have damaged spinal tissue, because those cells won't be working properly in them. So we can use those stem cells that we've made to turn them into nerve cells. Or people who are diabetic and don't produce insulin. Insulin is a very important substance in diabetes. Again, that links to a later topic. We can use stem cells to turn them into cells that produce insulin, and now that person who is diabetic will therefore be able to produce their own insulin. Now, there are various issues involving stem cell research, certain moral issues. Some people are a little bit against it because they feel that human embryos shouldn't be used for experiments. You need to be aware of these potential issues, and you might have to discuss it in a question as to why some people are against, or the advantages and disadvantages of it. Some people think obviously that curing patients who already exist and who are suffering is more important than worrying about human embryos and whether or not each one is a potential human life. Often they use embryos from fertility clinics, so when someone had an abortion, those fetuses would often be destroyed. So it makes more sense for us to use those stem cells if they can be used for a useful purpose. So just be aware that there are some moral and ethical issues around using stem cells. In some countries, actually, stem cell research is banned. In the UK, it's allowed as long as it follows certain guidelines. So just be aware that these are things that you may need to discuss. We won't go into them totally at the moment, but it may be something for a further video. We now move on to talking about transport in cells. So these three things that we are about to talk about, the diffusion, osmosis, active transport, these are very important to understand because they underpin a lot of what goes on inside cells, plant cells and animal cells. So we'll start with diffusion. Diffusion is, and you need to know this definition, it's the movement of particles from an area of higher concentration to an area of lower concentration. Often you can just think of diffusion as spreading out. If someone walks into a room wearing perfume, then that smell of that perfume gradually spreads around the room because it moves from where there's a lot of it on the person to where there's not a lot of it as it spreads around the room. So that is diffusion in the air as gases move from where there's a lot of them to where there's not a lot of them. Diffusion happens in liquids and gases because these particles in these substances are free to move around, so that the gas particles move around the air very easily. So here is an illustration of what's going on here. We have a lot of perfume particles over here. There's a high concentration of them there. They then gradually move to where there's less of them and they spread around the room. This happens a lot across cell membranes, and this is how substances often move into and out of cells. So cell membranes, as we saw earlier, hold the cell together and they allow substances to go in and out of the cell. Dissolved substances, so substances that are very small essentially, can move in and out of a cell by diffusion. So only very small molecules, as it says here, can diffuse through cell membranes. So oxygen as a gas, glucose because it's quite small, amino acids, again we'll come across later, water, these are substances that move into and out of cells very easily. A cell membrane, as it shows here, has got tiny gaps in it, so it allows small things through and larger things don't get through. So again, we have diffusion happening here of some of these glucose molecules. There is a larger concentration of them over here and they move that way through the cell membrane to where there's less of them. So that is how things get in and out of cells. Again, larger things can't. And that's why later we learn about digestion and how we break down these larger molecules into smaller ones to allow them to go into and out of cells. You also need to understand the rate of diffusion and why diffusion sometimes happens quicker in some instances and slower in others. So the rate of diffusion is affected by the concentration gradient. You are going to see this quite a lot. It's a fancy word for saying if there's a lot of them on one side and very few of them on the other, that diffusion will happen much more quickly. Again, using the perfume example, if someone comes in wearing a lot of perfume, then it will spread much more quickly around the room. Okay, it's kind of obvious when you think of it that way. So over here, we are going to have faster diffusion because we have got a very high concentration of a substance inside the cell, a low concentration outside it. And so this diffusion out will happen from a high to low concentration very quickly. Here, it'll happen much more slowly because we have a larger concentration outside the cell. And so it will still move high to low, it will just move more slowly. So that's why diffusion is affected by the concentration gradient. Literally, that just means the difference in concentration between the high and the low. Other things that can affect the rate of diffusion would be the temperature. When it's warmer, particles just move faster generally. So they will be able to move from one place to another more quickly if it's warmer. And secondly, the surface area of the cell membrane. We talked about this with the root hair cell, if you remember. So that root hair cell, which had the sort of extra long cell membrane, means that diffusion happens faster because there are literally just more entry points. So a cell like this, things can get in and out through the cell membrane, but if that cell, you know, has got a longer cell membrane or a larger surface area, there are more places for substances to move in and out. So you need to phrase that as a larger surface area. We now move on to talking about osmosis. Osmosis is very similar to diffusion in the sense that it is the movement of particles from a high to a low concentration. However, osmosis is specifically for water. Okay, so only water molecules moving from a high to a low concentration, and also the definition specifies across a partially permeable membrane. So unlike diffusion, osmosis can't happen through gases, for example. So for example, if you had a glass of water, it doesn't then spread out around, mainly because it can't get through the glass. So water doesn't just move around in the same way that gases do, and that's why there's a different definition for osmosis. So it only happens through cell membranes, and again, we have a high water concentration over here. We have the cell membrane, or the partially permeable membrane. Partially permeable just means it allows some things through it, not all. And so the water molecules move from a high concentration on this side to a low concentration on the right-hand side. So, as it says here, the solution surrounding a cell will usually have a different concentration to the fluid inside the cell. So that, as a result of that, water does always move in and out of cells. So the best example I can give you for that is if when you, for example, have a bath or in the swimming pool, often your skin looks a little bit different when you get out because water has moved in and out of the skin cells, because there is a different concentration of water surrounding those cells, and that happens throughout your body, and particularly in plants. Plants, when they're short of water, they will often wilt because water will move out of their cells into the surrounding air around them, and so that results in obviously the plant struggling when it is low on water. So as we can see here, if this is your cell, it has a low concentration of water inside the cell, a higher concentration outside it, so water will move in to the cell that way. On the right hand side, we have a higher concentration inside the cell there. Water would therefore move out of the cell. And again, there is a practical involving potatoes, which we will look at in a separate video. And lastly, active transport. Active transport is just the other way around. So it is the movement from an area of lower concentration to an area of higher concentration. The crucial difference here is that this doesn't happen naturally. Diffusion happens automatically, as I explained with the perfume example. Active transport almost needs to be forced the other way, so it uses energy from respiration. So any cells that need water or any substance to go the other way, so from a low to a high concentration, will need energy. And that again now links back to the mitochondria. Any cell that needs to use this form of cell transport, active transport, needs to have mitochondria to give it the energy it needs to force substances to go the wrong way or the other way or against the concentration gradient. So active transport allows cells to absorb useful substances even when the concentration of the substance outside the cell is very low. So even when you have substances outside, the more inside than outside, they can still take up more of the substances that they need. Now, there's only two places where you need to think about this for GCSE Biology. There is plant roots, because plants, the roots will need to take up as many minerals as they can from the soil around them, even if they've got a higher concentration of those minerals already inside the cell. They'll need to force those cells the other way. And also in the small intestine, cells need to be able to, if you've eaten a little bit of food, they need to be able to absorb that small amount of food, possibly from a low to a high concentration. So here is active transport in plant roots. So as you can see, inside here we have a high concentration of these mineral ions. Outside the root here we have a very low concentration, yet still, thanks to active transport, this cell is able to absorb these small amounts of mineral ions outside. In order for it to do that, remember though, it will need mitochondria in order to be able to get the energy that it needs to be able to do that. And then in the small intestine, or the gut as they often refer to it as in textbooks, it's used in the digestive system where there is a lower concentration of nutrients in the small intestine, but a higher concentration of nutrients in the blood. So we still need to get these nutrients out of the small intestine into the blood. So even though there's a higher concentration there, if we allow diffusion to happen naturally, diffusion would go the other way, and that's the wrong way. We want these things to be in the blood. So active transport allows us to take up a smaller concentration of these nutrients, and this, as it says, is essential to stop us from starving. Otherwise we would never be able to take out the small amount of nutrients from the food that we eat.